So thank you very much for joining us today. And so we're going to tell you about our research into marine plastic pollution within Narragansett Bay. So my name is Andrew Davis. I'm an associate professor at URI, and I've been working on marine plastic pollution for several years alongside my colleague, Professor Suckling from the College of Environment and Life Sciences. And today, what I'm going to present is kind of the broader research vision of URI. I'm also going to introduce you to the Land to Sea collab, which I'll explain in greater detail what that initiative actually is. I'm going to introduce our project team and the co-presenters today that are joining me in delivering the presentation. And we're also going to talk in more detail about our Sea Grant funded research program, focused entirely on microplastics within the Bay. So Dr. Suckling is my co-presenter and colleague, and Dr. Suckling will introduce microplastics, current knowledge and impacts, so the state of the current science, and she will also talk about what's happening in the state of Rhode Island. And Sarah, our PhD student, will talk in more detail about the research that we're doing with the focus of plastics in the water column within Narragansett Bay, and she will also present some of the future directions that we're taking in terms of our research program. And she's also going to touch upon how you might want to get involved with some initiatives and some other areas to maybe you can think about your plastic usage, for example. People are becoming increasingly interested in how they interact with the world around them. And plastics is one way. Every single day, we touch and interact with plastics in some form. Our food is packaged in plastics. Children's toys are often plastic material, and the clothes that we wear often contain plastic polymers. They're very durable, they're very useful, they're very valuable materials, but they do have some impacts. And that's what we're going to really talk about today, is what happens when plastic ends up in our natural system. Our research is being funded by a wide variety of different agencies and funding bodies, and I just want to highlight here some of the support that we've received, principally from Sea Grant, Rhode Island Sea Grant, which has really enabled us to kickstart our research program. So as you can tell, I'm not American, I'm British. So Colleen and I, we moved over to URI in 2018, and we had to rebuild our research program after closing it down in the UK universities. So it's been very grateful to receive support from all of these different agencies to help us rebuild our research program and really start, we hope, to make an impact on our knowledge within Rhode Island. So we have this beautiful sign, which we take out with us when we go field sampling and in various parts of Rhode Island. And we're really obvious, okay? Our students will be very, very obvious in terms of what they wear in the field as they're sampling. And Sarah will explain in more detail why exactly our students and staff are so vibrant in color, as it were, as we sample for microplastic in the Bay. But it's an increasing thing that we're doing. We're trying to get a really good understanding of the dynamics of plastic pollution within Narragansett Bay, how is it moving around the Bay, and where does it end up? So University of Rhode Island is deeply invested in microplastics and plastics research in general. There's several large areas of focus for URI, but one that is really coming through is research on coastal systems. We have Narragansett Bay, one of the most important area of water that we have in the state, and we're continually trying to work on this. So we highlighted plastic pollution during the recent fall honors colloquium, which you may or may not have interacted with, which was called Sustaining Our Shores. JP Walsh from Graduate School of Oceanography and myself were the co-conveners of this series of presentations from experts from around the world. And we had two major sections that focused on plastic pollution. The first one was a visit from Chelsea Rockman, who's a faculty member from Canada, who's one of the world leading experts in marine plastic pollution. And I've given you a YouTube link and a QR code so that you could go and watch the recording of Tracy Rotman's talk to learn more about what's going on in terms of the international and national level research into plastic pollution. We also had a panel of several solution-minded individuals talking about the impacts of marine plastic pollution, both at the state and regional level, which is on the right-hand side of that screen, November 30th. So these are two great resources if you want to learn a bit more about the discourse surrounding marine plastic pollution. And it was in Rhode Island. So Meredith said plastics land to sea and the URI collab. So the collab has been driven by the Vice President for Research, Peter Snyder, in a way of trying to bring multiple researchers to tackle a grand challenge, as it were. Okay. And plastics is one of these large global challenges that requires interdisciplinary, multidisciplinary research across many different sectors. And the first collab is focused on plastics, not just plastic pollution, but also material science, sustainability, a lot of questions surrounding, alternative materials and so on. So the Plastics Land to Sea collab, you will begin to hear more and more about this as this year goes by into next year, as it continues to build momentum. We were impacted quite heavily by COVID. We did have an initial meeting prior to COVID starting all of the lockdowns, but we kind of hit a speed bump. But it's bringing back up to speed again today. So keep an eye out for the collab, because we're going to see a huge amount of research focused on plastics coming from URI over the next several years. It's really trying to bring people together, researchers together, communities together. It's really thinking about solution-focused science. And it really starts with that whole point about plastic textiles, our use of these materials. We need to think about the tools that we have to work with them, the behaviors that humans have in terms of social science, or how we interact with these materials, what impact do they have on our natural system, and how we can start to solve them. So if you are a researcher or you are a stakeholder and you are interested in learning more about plastics and about how URI is moving forward, there is the Plastics Land to Sea website, which is got a lot of information on, a lot of contact details of different researchers that are interested in this area of plastic research. And also we recently had the Research Momentum magazine was published, which was focused entirely on plastics. So there's a lot of resources starting to be developed at URI that you can start to interact with. So what I'm going to do now is, after that brief introduction, is hand you over to my colleague, Dr. Suckling, who's going to introduce microplastic current knowledge and impact, and give us a little bit more of a focus on what's happening in Rhode Island. So thank you very much. Dr. Suckling, please take it away.
Thank you, Andy, for that great overview. So just please give me a second while I share my screen, and then I can get started. So I'm hoping that people can see my slide okay. Can I get a confirmation? Perfect, thank you. Well, this is, I want to say thank you to everyone who's joined the session today. This is really exciting to be part of this community and sharing a bit more information about what it is that we've been up to here in Rhode Island, and how we can help sustain our oceans and the community of people, and learn more about how we can have impact with the research that we're doing here. So I also teach a marine plastic pollution course, which is a relatively new course, a grand challenge course here in the University of Rhode Island, to kind of educate students about the kind of emerging topic that is plastics. This is a new area of science, what is garnering a lot of international interest and focus. We have a lot of questions that we still don't have answers to. So we're hoping that we can help contribute towards untangling, what is the issue that is plastic. And the first slide that I always start with my class is asking, what are plastics? And students quite often really struggle to really define what a plastic is. And this is a kind of general description that I give to everyone about plastics. They are synthetic or semi-synthetic materials And when we say synthetic, what this means is that they're actually made from fossil fuels. And this is something that often people don't realize. It's something that I didn't even realize until I started working in this research sector. So basically, when we have oil refinery processes, we're basically breaking down crude oil into lighter hydrocarbons that they're easier to use across the commercial market. And plastics use one of those byproducts as well. So they are made from fossil fuels. And as many of you can imagine, this is contributing to some issues towards climate change as well, because of the effects that fossil fuels can have on our atmosphere. And when we use this term semi-synthetic, this kind of like starts getting interesting, because we can combine these fossil fuel kind of based kind of components and mix in natural products like plants, like cellulose, for example, to kind of build towards plastics. So some companies have been using this term bioplastics to produce plastics which may offer alternative routes to plastics, but it doesn't necessarily move us away from this use of fossil fuels, unfortunately. So these like synthetic and semi-synthetic materials made from fossil fuels. Now, we love this stuff so much. Let's just look around all our offices now where we're kind of zooming in from. What can we see? We can see headphones, we can see computer keyboards, we can probably see carpets and things. These all have plastics in. Even the paint on your wall has plastic in. It's everywhere. It's amazing material. It's resilient. It can be built into anything that we want it to, and it's very affordable. So our use of it is not really going to reduce that much. It's everywhere, the plastics. And in 2017, our kind of last kind of really average, our last kind of snapshot of our detailed use of it, we produced 350 million tons of plastics in that year alone. And that is only going to increase. Pre-COVID, the trajectory of increase was that it was going to triple by the year 2050. Post-pandemic, with the pandemic taken into account, we may see that number increasing, because we've probably all experienced and seen the increased use of single-use plastics as a means of reducing the spread of COVID by this kind of like throwing away these plastics to protect everyone. So we're probably going to see this increasing. Now, when we look at an ocean perspective, everywhere that we have sampled pretty much so far, we have found plastic. Whether it's the ocean surface, the deepest part of our oceans in the Marianas Trench, we have found plastics. Even in the remotest areas of the world above mountaintops, Mount Everest has had found plastics on there, and even in Antarctica and the Arctic. So nowhere is untouched by plastic, unfortunately. And a lot of this like plastic that we're using, this schematic shows the kind of increasing trajectory of plastic and how we are basically just increasing like climate changes, the use of plastic. But what is really concerning is the kind of the waste that this plastic production and use creates. We have a huge amount of waste, and unfortunately, much of that is mismanaged. And I'll talk a little bit about that in a few slides' time. But so far, we have an estimation that 8 million tons a year is entering our oceans. So that is a problem, because obviously the oceans cover a huge part of our planet, and this is a material that does not belong in these oceans. So with the increase in production of plastic, the increase in its use, plastic waste going into the oceans is only going to increase as well. And when we start looking at what kind of, how much of the debris, this is like the non-natural materials which is found in the oceans, is plastic, the vast majority is, 85% of what we find is plastic, unfortunately, what we're finding. So it is a kind of substantial problem. So when we look at plastics, especially in the research sector, we kind of categorize them or define them by their size, because they come in all shapes and sizes, as many of you will know. So just to kind of put it into kind of simple terms, we call macroplastics these large plastic particles, anything that's larger than 5 millimeters in size. So this will be anything that you find on the beach, for example, or maybe large items floating in the ocean. And then we also have microplastics, which is a term I'm sure many people have heard about beforehand. And these are smaller microscopic sized plastics that range from 5 millimeters in size down to microns in size. So smaller than what the naked eye can see, unfortunately. And you can see they come in kind of a range of shapes and sizes, these fragments. We have these spherical beads. Some of you may have seen these on the beach. These are called nurdles. These are kind of typically how industry will create the first stage of plastic in its lifetime in these small spherical beads so that they can be easily melted into whatever it is they're going to be created into, like chairs, furniture, whatever. So we have these like small nurdles which starts entering the chain of plastic production and use. Now, also, we have the problem where the two are kind of related in the ocean when it comes to pollution and waste. So basically, if we have a large or macroplastic that we find in our ocean, so we have this single-use plastic bottle here, for example, over time in the ocean, this is going to degrade very, very slowly, because remember, these are resilient materials. So we have the UV radiation from the sun will weaken its structural integrity, combined with the kind of physical forces of the ocean and friction, and this bottle being bashed around by waves and hitting rocks and other hard items. Eventually, over time, this is going to break down into these smaller pieces. So your macroplastics can actually fragment down into microplastics, which can be an issue. But there are many ways in which plastics can actually enter our ocean ways. So here I'm just going to name some of the few kind of main sources that we know about, but this is not a fully inclusive list here. It's just some of the major areas that we are aware of at the moment. So one of the major ways that we know about this is through sewage. We have these sewage-related debris where we have these small beads in some of our kind of beauty items that we have in our bathroom. So anyone who has face scrub or maybe whitening toothpaste, you may be horrified to find out that some of those have or may even still contain these microscopic plastic beads. It sounds kind of gross, but these things we use these to abrade the kind of surface of the skin so you get beautiful skin afterwards. But we wash that off in the sink in the bathroom, and then this goes directly into the sewage. Same for domestic washing. If you're washing your textiles, like your bedding, your clothing, a lot of microfibers, especially from synthetic textiles like polyester, for example, will wash off from that washing machine load and go into wastewater, the sewage. And sewage will generally go to wastewater treatment centres where there is a whole process that goes on to remove solid materials. So they may float materials out, they may sink them out and remove that sludge, and some of those plastics will be removed with that process. But eventually that processed water will enter the aquatic environment, whether it's into estuaries, into kind of river areas, but generally coastal areas. This is where the wastewater that's been treated will end up. And it ends up being passed through its final stage through a small screen, which is only about 1.5 to 6 millimeters in mesh size. So this means that it's not able to capture and isolate many of those microplastics, so they end up coming into our ocean. We can't really realistically put smaller mesh size screens in because they're just going to be continuously blocked. So people are looking at ways to try and improve this intervention to reduce this, but at the moment it is one of the kind of major sources of plastics going into the ocean. Another major source is obviously mismanaged waste like litter, for example. So we may find it on the beaches or in city waste, or like overflowing trash cans, or people leave it behind. And these will get blown or washed with rainfall into rivers or even into stormwater drains. And many stormwater drains just go directly to the coastal environment. They have no intervention for removing particles at all. So mismanaged litter will ending up in our waterways that way. Accidental loss through industry as well. So remember those little like tiny nurdles I mentioned, they can end up accidentally into the ocean. Even equipment that's lost, like containers off container ships. We had an incident, I think, over the last year where near the coast of India, a container containing millions and millions of these nurdles was released into the ocean and ended up in the coastline in some parts of India. So that inundated their coastline. And then also we have some lost marine equipment as well, like ropes, nets, anchors with rope attached to them. These can end up being lost at sea, and we've heard about perhaps the term ghost fishing, where some of fishing equipment will continue to function to capture fish, for example, as it's moving along. So they can be quite impactful, unfortunately. So when we look at domestic waste, there have been routes to try and reduce some of the the microplastics entering our ocean ways. So in 2015, we had the Microbead Free Waters Act that came in. And what this does is it prohibits the manufacturing, packaging, and distribution of rinse-off cosmetics containing plastic microbeads. So it's moving in the right direction, which is great. But notice that language, it is saying rinse-off cosmetics. So it's very selective in terms of the products it's targeting. But there are many products out there that still contain microbeads. So it's a step in the right direction, but we perhaps need to do a bit more. And with this domestic waste, we also still have this issue of like fibers coming in through the laundry. So scientists based in the UK, they have a lab with a scientific washing machine where they basically did a bunch of washing with textiles with acrylic, polyester, and polyester blend cotton. And they measured how many fibers were shed from these particular types of materials in your average 6 kilogram wash in a washing machine. And I think you'll agree with me, this is kind of disturbing. With acrylic washing, we had almost 750,000 fibers being shed in just one single wash. Polyester, we had almost 500,000. And these polyester cotton blend, even though it's less, there's still hundreds of thousands of these fibers being shed per laundry wash, which is going straight to our wastewater and into our oceans. And when we look at those fibers, fibers form a large component of what we're finding of microplastics in our oceans in general. So this isn't a Narragansett-based specific example here, but this is just a general understanding of where things stand in a global average. So synthetic textiles are forming about 35% of the microplastics that we're finding, these fibers in our oceans. We're also getting, this is something I didn't realize until I started working in this field, car tires. So car tires are made of 50% natural rubber and 50% synthetic rubber. And I guess it kind of makes sense because, you need to build in extra materials synthetically to make the tires be very resilient for road use and have like high wear. But these are friction against the road and small particles are falling off the tires and then being washed into the coastal regions. So 28% of what we're finding in our waters comprises of these car tire microplastics. And then another quarter of what we're finding is city dust. So this is basically air carrying weathered, abraded, human-made products across the city, which can include fibers perhaps from dryers. That's received some interest in media recently where the kind of fibers or lint coming from your dryer is going out into the air, contributing to this issue. We also have very good sort of resilient paints, which we're using on roads, but also as marine coatings on ships and boats. Over time, these are breaking down and contributing toward to up to about 10% collectively in the water. And then smaller components are coming from this like industrial sort of nodal constituent, and then also from personal care products. So we know microplastics are in the water. We found them, researchers internationally have found these microplastics wherever they have been basically sampling across the world. So the kind of like next step really is to understand coastally, like what, we trying to identify where those kind of sources are coming from for unique kind of coastal regions, but also trying to predict and anticipate where those particles are going so that we can potentially identify any vulnerable ecosystems or areas, or to help us identify any hotspots where we can maybe start intervening from a waste management perspective. So to do that, we have to start looking at the physical characteristics of the plastics. And this is something we also use in our lab work as a way of isolating them from seawater. And Sarah's going to talk a little bit later on about what that looks like. But when we talk about the physical characteristics of the plastic, I'm talking about its density. So anyone who's not familiar with this, what I mean is the mass per unit volume of an item. So what we're talking about is grams per centimeter cubed or cubic centimeter. Okay. So when we have a plastic, if we want to know if it floats or if it's going to sink, we have to know what its density is relative to fresh or seawater, for example. So fresh water has a density of 1 gram per centimeter cubed. And seawater is a little bit denser because it has all these salts and minerals in it. And it has a density of about 1.02 grams per centimeter cubed. And it's represented by this thin blue line here in our kind of sliced water column picture that we have here. So anything that's any plastics that are going to float, they will typically have a lower density than seawater. So you have things like bottle caps like polypropylene, or plastic bags, which are made of like typically polyethylene, or even expanded polystyrene or styrofoam, which is that kind of plastic with lots of air fused into it. And then any materials that are not going to float and they'll start sinking will have a heavier density. So things like fishing nets made of nylon, perhaps these like cigarette filters that people may drop at the beach made of cellulose acetate, and also things like polyethylene terephthalate like PET, like made of whole drinking bottles. Now it starts getting challenging because when we think about some of these materials, like bottles, for example, if the cap's kept on, it's got air trapped in it, it's going to float. And then until that surface breaks, it will then become entrained with water and start sinking. So that makes our job of predicting where plastics go a little bit more challenging. But then so does the incredible biological processes that happen in the ocean. So something that we call biofouling, some people may relate to this, where they may have had a plastic material that's been sat in some sort of water for some time. Then you kind of get this like horrible kind of like slimy residue on the surface of the plastic. That is what we call a biofilm. It's bacteria. It may even be some like microscopic plankton that's building on the surface of your plastic. Now, what's really challenging to know is what rate of biofouling can occur in a plastic to enable us to predict where those plastics are going. And we still don't have answers to that question because different plastics will respond to biofouling in different ways. It also depends on how productive your coastal regions are, and coastal regions across the world are very, very different. So that makes our job much, much harder. And at the same time, when biofouling does occur on a plastic, when it's being pushed around in the ocean, it changes its density, making it heavier, more dense, therefore affecting its point at when it starts to sink in the water. And all coastal oceans are very dynamic. They are moving around all the time. We have currents, we have tides, we have storms, which basically move water at greater distances and greater capacities. We may have like interesting profiles of the seabed as well, which will affect how your water moves around. All of these make it very challenging to try and predict where those go. I know here in the University of Rhode Island, we have a really good grasp and understanding about what water movement looks like in Narragansett Bay. So it puts us in a really good position to start thinking about these questions. And there are some researchers here who are looking at the behavior of these plastics relative to this biofilm. But we still have a lot of answers that we need to address around this. And this schematic here, this just really highlights some of those processes that happen as an example. So you get your plastic coming into the river, it gets beached on the beach, and then it gets washed off and pushed around by wind currents at the surface of the water. There'll be some turbulence going on, which will disturb it a little bit. And then eventually it'll start biofouling and sinking towards the seabed. And the eventual fate of most plastics in the ocean is to sink to the seabed. And that means then that the seabed is going to be a major site for receiving a lot of this plastic pollution that's coming into our oceans, unfortunately. So we're going to go back up to the surface of the ocean and think about those floating materials. So we know that plastics that are light or have low density, they're pushed around by wind, they can be distributed at very, very far distances. So we have these kind of like trade winds which push the surface waters across our planet around, and where they start colliding, we get these very kind of like slowly spinning kind of like gyres in these areas. And what happens is plastics that are being pushed along by these winds get trapped in these gyres, to the point where we have this, well, some of you have probably seen this, the Great Pacific Garbage Patch, one of these gyres in the Pacific Ocean. And we have huge amounts of plastics that can collate in these areas. So when this Great Pacific Garbage Patch was found, people were identifying its scale as being so big that it almost was like a landmass. It was how extensive it was. And some estimations showed that there was about 80,000 tons of buoyant plastic in one of those gyres, mounting to about 1.8 trillion pieces of plastic. So it was very extensive. Since the discovery of these, there has been huge international cleanup efforts to remove those. But it does create some issues of like microplastic presence, because these materials have been breaking down and colliding into each other. So we're not quite sure how we can really deal with that. But what this really emphasizes is that this is a global scale issue. It's everywhere. And you think about planet Earth, over 70% of the surface is covered in water. So this is extensive, and this is why we are worried. So plastics being pushed around do provide sort of risks to our ecosystems for various reasons. So firstly, we can see that marine animals, so here we have something like goose barnacles, can attach very well to the surface of plastic, and they will be distributed around the world. And this creates a risk of introducing species which are not native to an area, making them invasive. And there are many examples across the world where invasive species have come in and pushed and competed out more sort of native species. So it can create risks for these ecosystems. The Japanese earthquake, which happened, I think it was around 2011, which was induced a tsunami in Japan, that pushed a huge amount of material, plastic debris across the global oceans, so that months and years later, Hawaii were actually receiving a lot of the plastic materials on their shoreline, and many of them came with invasive species that could have endangered their ecosystem. So they're now monitoring their beaches with regularity to prevent this from happening. Other risks that plastics can cause as well is the ability for bacteria to, like these biofilms, the bacteria can form on the surface of them. And in the Pacific, in coral reefs, they have found Vibrio, which is a very bad pathogen on the surface of plastics, and they get entangled on these like corals which cannot move anywhere. They are, these static organic objects. And what happens is they abrade the surface of the corals and risk their uptake of the pathogen, which can cause a disease called white syndrome, which basically destroys the coral tissue. And what researchers have found is when these corals come into contact with these plastics, it increases their risk of this disease from 4% to 90%. So there is a hugely a huge increase in risk of this disease uptake, unfortunately. So again, there are cleaning efforts going on at play, but there is this risk to ecosystems. Many plastics are built with chemicals in them to serve certain functions, like to be antimicrobial, which is very popular with the COVID at the moment. Some of them will have anti-flammable properties as well, which requires chemicals to be built in. So that also presents a risk of like chemical exposure to animals potentially, although we don't understand this very well. So it's something that researchers are looking at more closely to get more answers. But Rainer Lohmann in Graduate School of Oceanography has shown that some plastics can accumulate some persistent organic pollutants onto the surface of the plastics. So things like polychlorinated biphenyls, PCBs, is a good example of this, where they can build up on the surface of the plastic. And then we know that animals interact with these plastics and ingest the plastics. And if that is passed from one animal up to the next animal in the food chain, those PCBs can build up the concentration, so they bioaccumulate across the food chain. And PCBs have been shown to be very detrimental, very bad for a range of marine animals, very toxic. So people are concerned about this and are looking at this more closely to really truly understand what that risk poses. But this also includes other things like polycyclic aromatic hydrocarbons and also DDT, which some people may have heard of. So we have this chemical risk, but what about the plastics themselves? We know that they're resilient. That is one of the appealing properties of these materials and why we use them so widely. But unfortunately, that means that their breakdown in the water takes a very long time. So fishing line is estimated to take about several hundred years to break down. Plastic bottles, again, several hundred years to break down. So what this really highlights is not only do we have a global scale issue here, but we also have a long-term issue. And when we start thinking about other issues at play like climate change, we really don't know much about how these two are going to be interacting with one another as a risk to marine ecosystems. So it's something that our lab is starting to look at and address. So what is the current state of the field then in terms of like plastic pollution, especially with microplastics? Well, we know that microplastics are found everywhere in the ocean environment. They're commonly found within many marine organisms. Pretty much any animal that has been dissected, we have found microplastics, unfortunately. So what really we need to determine is what extent do the microplastics, if any, have an impact on those marine animals, and also trying to understand a bit more about how they travel through food webs. Now, I'm going to show some examples of like how animals are interacting with plastics, but there is a warning here if there are some pretty disturbing images I'm about to use. So please look away or close your eyes until I say it's safe to look again if you don't want to see these. So I'm going to move on to the next slide now. But what we have is basically a lot of entanglement of plastics. We've seen fishing line, we've seen frisbees have shown as an emerging problem as well, where seals will play with the frisbees, they'll flip over their head, and then they get stuck there and they can't remove them, and they grow, continue growing with these frisbees attached to them and digs right through and penetrates their skin. There has been some mortalities, unfortunately, associated with this, and there's a lot of like rehabilitation like programs running in the UK to help rescue seals and remove these and make sure that they have a kind of like safe recovery and then are reintroduced back into the wild. These are the stomach contents from a sperm whale that washed up on the west coast of Scotland some years ago. There is a system there where there's a marine mammal like organization where they will do an autopsy on the marine mammals so they can learn a bit more about what the impacts are like on mammals and how they've died, to learn a bit more about what kind of management is needed in the ecosystems there. But the prognosis for this sperm whale was that they had died from artificial satiation, meaning they were full of all this marine debris here, so it's ropes and nets, and they were unable to eat anything else. So it basically filled them up artificially and meant that they were unable to process any more food. So, you know, this is a pretty, we are seeing more presence of mammals containing these kind of like materials because they mistake them for food items. They can't differentiate them visually from food. So this is definitely one of the reasons why we need to reduce plastic pollution coming to our oceans. So the warning's over. It's safe to look back at your screens if you decided to look away. So we have like sampled internationally as a research community. We have sampled a whole range of different animals: fish, shellfish, crustaceans, plankton, seabirds, worms, snails, sea squirts, marine mammals, just to name a few. And we have found plastics, microplastics in those animals. In many cases, we are seeing a very extensive presence of fibers in those animals. And this brings me back to the earlier point where we're seeing a lot of fibers coming into our marine environment, and it forms about 35% of what we find in our oceans. So animals are interacting, therefore, with these plastics. Maybe it's through accidental ingestion, whether it's through inhalation whilst respiring underwater, I don't know. This is something we're trying to elucidate. But there are also some like presence of these fragments and these beads as well. So they're there in animals. Now, researchers are trying to understand what the impacts are like. Well, we know that the presence of plastics are there in the animals, but one of the questions we're trying to ask is, how are those animals being impacted? Does it affect their condition? Does it affect their physiology? And to do this, we have to conduct a lot of lab work to simulate and expose those animals to those plastics so that we can understand how their physiology and their condition has been impacted, if at all. So anyone who may have seen media articles about microplastics and their impacts on marine animals probably have seen a lot of like doom and gloom reports, especially when it comes to microplastics. So I want to kind of like just highlight that, you know, we need to take a step back from what that means, because when we look at those lab studies, a vast majority of them have utilized very unrealistic types or concentrations of microplastics in these kind of artificial lab conditions compared to what we would find in the natural environment. So they've used concentrations which are by several orders of magnitude much greater than what we would find in the natural environment. So there's a bit of a mismatch at the moment in terms of what we know is an environment, but what the impact is of those concentrations. These kind of high concentration studies are important to get initially so that we can understand the direction some of our questions may need to go. So, you know, what happens if we give this animal a lot of plastics, and what is the response that we can measure? So what does this look like in an environmentally relevant context? But unfortunately, we don't have many studies yet which have looked at environmentally relevant concentrations or types of microplastics to understand what that really truly looks like from a management perspective. And this is something that our lab is doing, which Sarah will talk about in a moment. So I'm nearly done. I'll hand over to the next person soon. But here's an example of a marine animal that sits on the seabed, which is the eventual fate of many of these plastics that sink through the water. We have a sea urchin, and it has these, it will move along the seabed, and you can see it has these teeth, and you can see them opening wide and then closing and then drawing food into its body. So they're constantly grazing on the seabed and ingesting plastics potentially. And I did a study in the UK where we looked at providing microplastics at environmentally relevant concentrations to two types of sea urchin, one that feeds on a lot of like soft algae materials, so it's not used to ingesting lots of hard parts as part of its diet. And then there's other species which feeds on soft algae, but also feeds on small animals which have skeletons and shells, so it has hard parts of its diet coming into its body with regularity. And what we found was that we see increasing sensitivity to microplastic ingestion when animals are much more used to a kind of soft diet intake. So this gives us some insights that dietary habit might be a good thing to look at as a way of understanding how sensitive species might be to plastic exposure. And just recently, a graduate student, Hannah Haskell, has done a study by looking at fibers which we find in the marine environment, polyester fibers, and exposed the eastern oyster to these fibers using environmentally relevant concentrations. So what those look like is basically we can have anything between 0 and 1 particle per litre, all the way up to about 95 particles per litre in very sort of densely populated city coastal waters. And then we had a really high concentration here where we had 950 fibers per litre. We use this because this is a concentration which is commonly used in laboratory studies so that we can see how well this study was comparing to other work that was done. And overall, with the things that we measured, things like how well they were good at clearing water with microalgae in, they seem to be relatively unaffected by the presence of these fibers. So I found these results very encouraging. We need to examine this a little bit more closely to see what this is like for other types of plastics, but we've certainly found this very encouraging. So what is Rhode Island doing to tackle plastics? Well, Governor Raimondo, when she was governor of Rhode Island, signed an executive order to tackle plastics. So she got a whole bunch of people from across the state to understand where we can try and reduce our reliance on single-use plastics. And they've come up with this excellent recommendations report, which they're now starting to work on. So we can provide this link later on if need be for anyone who might be interested. But there are many organizations which are working on cleanups and education and awareness with plastics. So we've got Save the Bay and Clean Water Action and Ocean Access. They do like beach cleans, and they will also use some information about plastics to lobby for changes towards to reducing plastic pollution. Roger Williams University is also looking at microplastic impacts on marine animals. And also the EPA, the Environmental Protection Agency, has been measuring plastics and sediments around Narragansett Bay, and we can talk a little bit about that later. But overall, there have been bans on in some regions of the state banning single-use plastic bags. And about 2-3 weeks ago, the Senate approved a legislation for a statewide single-use on plastic bag ban, requiring stores to offer recyclable options as a way to reduce single-use plastic reliance. So if this passes into law, then this could go into effect by January 2024. Anyway, I've taken far too long, so I'm going to hand over to Andy next, but thank you very much, everyone. I'm looking forward to hearing your questions.
Andy, you're muted.
Thank you. That changed my screen. The mute icon went really small and disappeared. So thank you very much, Colleen, for that introduction to plastic. So what we're trying to do now is hone down a little bit more into our research that we're doing, and particularly within Rhode Island. So we're going to talk a little bit about our Sea Grant funded research program, and I'm going to pass you to Sarah in a few moments. But what we're trying to do here is use Narragansett Bay as a model system. By that, what we mean is this is such a really well-studied, highly modeled, highly sensed, really well accessed site that we can get out there and do really good science. We can take our boats, we can collect samples, we can travel up and down the bay. And because of that really great access, it allows us to take advantage of what's going on. For example, we can start to look at the distribution of humans throughout the bay, look at north to south urban gradient, where down in South County, we have fewer people than in the north. We have an incredible watershed where water is moving into the bay. So there's lots of ways that we can start to think about the plastic pollution problem in Narragansett Bay. We also have really great networks of stakeholders and highly invested people who are working on the bay. And that really gives us additional power to start thinking about some of the things that we want to do in terms of engaging with stakeholders and potential users of the research that we do. So it's an amazing site to work in. So we came up with the Aussie Map project, which is what we're calling the Ocean State Initiative for Marine Plastics. And we have a website that we're working to try to keep up to date and to provide up to date information about what's going on in our research, but also within the research of our collaborators as well. So you can check out that resource if you're interested in plastic pollution in Rhode Island. So our project is based around 3 different objectives. Number 1 is that fundamental question that Colleen was saying was, what's out there? How does it change through time? So we're looking at this north to south gradient across Narragansett Bay. Objective 2 was to start to bring in this work that Hannah Haskell did and recently graduated on her master's program to look at what's going on with plastics and how it interacts with oysters. And then our final objective is to look at what's going on with food webs. Can we see if particles are moving through marine food chains? And in our example, a model system of oysters and crabs. So on that note, what I'm going to do is hand you up to Sarah, who's going to tell you about the research that she's been doing and some of the future plans and a little bit more about how you might want to get involved with plastic research.
All right. Thank you very much, Andy. I'm going to share my screen now to take over. There we go. Awesome. Hello, everybody. As Colleen and Andy mentioned, we are really interested in taking a multifaceted approach to understanding microplastics in Rhode Island's coastal waterways, especially in the Narragansett Bay. So I'm going to dive into some more detail on the ways that we are accomplishing those research goals. So that will include some methodology, both in the field and the lab, some of our preliminary outcomes, and plans for the future. So we currently have 3 main projects in the works right now to better understand our Narragansett Bay ecosystem. The first is a seasonal assessment of surface water debris, and this is using a manta trawl approach and a pump filter system sampling approach. We also have a project assessing our state's freshwater resources, particularly as a means of pollutant transport, and this is also using our pump filter system sampling. And then the third is better understanding movement of microplastics in the food web, interactions with organisms, especially through trophic transfer. And this is going to be undergoing some experimentation in URI's mesocosm facilities. So I'm going to start by jumping into our manta trawl surveys. So as you can see here on the map, we have 6 sampling sites that are spaced throughout Narragansett Bay, and we're sampling each of these seasonally. So we want to produce a really robust and technically sound approximation of the bay's pollutant load. So the spatial kind of organization of these 6 sites is really allowing us to not only sample from a north to south, kind of urban to less urban gradient. So we have Providence up here at the top, down towards the open ocean, but also the east and west passages of Narragansett Bay. We know that the east and west parts of Narragansett Bay are certainly not a mirror image of each other, and there's a lot of different dynamics going on in the bay: tides, currents, wind, rain, runoff from all these different land masses that could be potentially influencing the spatial spread of microplastics throughout. So how are we doing this? And maybe what are we starting to see? So in the field, I'm going to go through some of our techniques and then our lab processing as well. So we have a manta trawl net here visible in the middle picture, which is a 330 micron pore plankton net, essentially affixed to a stainless steel frame that has wings that allow it to float and basically skim the surface of the water as it's trawled behind a vessel. So water enters the mouth of the net and is pushed towards the back. So it's pushing all the debris that it's catching into the cod end, which is affixed to the very end of the net. So we use a URI research vessel to trawl in each of our research sites for 10-minute increments. We trawl against the current and we measure the total volume of water sampled using a flow meter, which is affixed to the mouth of the manta net. So we have really strong contamination controls on board, which Andy mentioned briefly, including wearing our beautiful orange jumpsuits. And the purpose of that is really to make sure that everything that we're collecting in our environmental samples is absolutely coming from the sampling that's happening in the water. So we have a couple of different controls in place to help us make sure that we are accomplishing that goal. The first is that we use filtered seawater on board. So on the boat, we have a gas-powered pump that is equipped with a 1-micron cotton filter that allows us to produce filtered clean seawater when we're washing down the manta net and when we're washing the sample from the cod end of the net into our sample jars. We wear our orange. The orange is cotton. So if we do ultimately accidentally get any fallout from those orange jumpsuits into our sample, it's not going to be ultimately selected in our final processing because it is cotton and not, for example, polyester, which is really important. Orange is also selected as our color because it is very infrequently found in environmental samples in testing for microplastics. So we trawl for 10 minutes, we pull the net back on board, we wash it down from the outside with our filtered seawater, everything goes into our glass jar, and that's our environmental sample. Between replicates, we also thoroughly wash down the net, another contamination control, and take what we call a net control sample. This allows us to potentially recover any extra plastics that might be hiding out in there so that we can make sure we know where everything is coming from and attribute it correctly. We also take an air control sample whenever our environmental samples are exposed to the air. So if we do catch any, orange fibers in those air contamination sample jars, we'll look really closely in our sample to check and make sure there's not any residual orange fibers in there as well. So we have all of our sample jars, all of our awesome, amazing Mantitrol work is done in the field. What happens next? We take it to the lab. So once our samples are in the lab for processing, we start with sieving to remove all of the excess seawater. So as you can see, we sieve all of the stuff down. We've got a lot in there. We have biological material. We likely have microplastics. We might have some other materials as well, like sediments, things like that. So we need to be able to filter through what is plastic and what is not. And in the Mantitrol technique, because our pore size on our mesh is fairly large, I know 330 microns, so less than 0.5 millimeter, does not seem large, but in terms of microplastics, it is on the larger end, and it is visible to the naked eye as well as very visible under a microscope. So because of that, we actually are able to hand select our particles from our Mantitrol work. Once our samples are sieved, we suspend them in a hypersaline solution. So we use supersaturated salt water with a specific gravity of 1.2 grams per cubic centimeter. If you're familiar with microplastics methodology, you might think that's a little bit low, especially when we saw the categories of densities of plastics. But if we remember that we're only sampling surface water with the manta net, we're really already selecting for those floating plastics. So as we're picking out these samples, we're suspending them in something that's a little more dense than seawater to help those plastics float up to the surface of our petri dish, as you can see here. And we're not expecting to see a lot of those denser plastics in this sampling work. So this project is really looking at what is interacting with kind of the top maybe meter or so of water that the Mantitrol is able to collect. So we hand select particles. Myself and our wonderful undergraduate assistants will sit under the microscope and pick out all these things that we suspect are plastics. This is not random. We use some characteristics that have been developed from the literature over time, things like color, shape, texture, density if they're floating. And we pick out everything that we suspect to be plastics. And I'll show you a picture kind of up close what that might look like in a moment. All of our suspected particles are then stuck to a grid on slides lined with double-sided sticky tape. And I'll go into that in a moment as well. And then every particle that we collect is imaged on a high resolution image using our Olympus BX63 automated light microscope. So we have a really high resolution image of every single particle that comes through our processing pipeline, which is really important because we want to capture those characteristics of those plastics. But the next step that I'll describe in a moment is slightly destructive. So before we undertake that, we want to make sure we have this original particle that we've recovered from the bay. So our grid system is really, really useful for making sure that we can track every particle from the moment it's picked out of that sample to the end of our processing. And the way that we do this is by lining glass microscope slides with double-sided stick tape. We have 5 rows, and there's 5 potential spots on each row for a particle to sit on. So this way we can give them each a letter assignment. So as you can see kind of in the mock-up here, if a particle takes up more than one of its assigned like letter spots, we'll just assign it multiple letters, which is very useful. So this way, if we wanted to return to a particle later, if I said I wanted to take a closer look at, for example, the 4th particle on this slide that's photographed here, and it came from our Beavertail Lighthouse site, this one might be called Beavertail Lighthouse Sampling Event 4, Replicate 2, Net Sample, Slide 4, Particle D. And it seems a little bit crazy, but having this catalog this way really allows us to have a very like well-organized system, which I think is really cool. Andy thinks this is genius. That's how he's described it, which I agree with. So the last 2 steps of our mantitol sample processing are really the steps that are allowing us to decide whether or not these particles are plastic or not. So this is where we can really make that distinction. And the first step is a hot needle test. So we poke basically every single particle, as long as we can be sure that we can leave a little bit of remaining unmelted particle for Raman spectroscopy, which I'll talk about in a moment, with a fine tip soldering iron. And the soldering iron is heated to 320 degrees Celsius, which is well defined as a threshold in which plastics will generally, for the most part, melt, and most other materials will not. So this is a great way to pretty quickly identify if what we've recovered is melting. We can have with strong confidence say that it's likely a polymer, likely a plastic. And if it's burning or combusting, we can pretty strongly say that it's not. So we can rule it out of our processing pipeline. The last step is ensuring we know the material type of what we've collected. So that is using Raman spectroscopy. And Raman is a really neat technique. It is one of 2 of the most commonly used techniques to identify microplastics from recovered samples, the other being FTIR, or Fourier transform infrared spectroscopy. And both Raman and FTIR are what are called vibrational spectroscopy techniques. So these are using light to shine these photons. We use a laser, a monochromatic laser, shine photons at our little sample, our tiny little piece of plastic. And what's happening is some of those molecular bonds are excited, which causes vibrations, and the photons that are returned have slightly different wavelengths. And that's collected by the laser and basically assembled into the spectrographs. We can see an example of that here. So this phenomenon is called a Raman scattering or Raman shift, and it can give us basically a fingerprint of each of these different types of plastics and lots of kinds of materials. But because plastics and polymers are primarily carbon and hydrogen, they have really distinct bonds. They're a great candidate for using Raman spectroscopy. We use Raman as opposed to FTIR just because we prefer it. It really is an arbitrary choice at some point. It's non-destructive. It is time consuming, which is why it necessitates subsampling. So we do not test every single particle. And when I tell you how many particles we've collected, you may understand why. But it does give us a very strong idea of what types of polymers we're collecting from Narragansett Bay. Once we have our spectrograph collected from the Raman, we can match it against a known spectral library. And these are really useful for matching lots of different kinds of materials. We've actually created our own custom spectral library. We created ours from the Polymer Kit, which was developed by the Hawaii Pacific University Center for Marine Debris Research. One of the reasons why this is really important is because it allows us to basically calibrate our matches to the nuances of our confocal Raman machine at URI. And it creates better circumstances for higher match confidence, which is really helpful. So that's our mantitol pipeline, our field work, our sample processing, and that is the research project that we've accomplished the most with so far. It's been ongoing for about 1.5 years, and I'll share some of those results in a moment. But next I'm going to talk about one of our really exciting methodologies for some additional field sampling that we've pretty much just wrapped up developing the processing technique for, and we're going to start implementing probably within the next month or so. And this is our stainless steel pump filter system. So while our Mantitrol project is great for assessing what we consider like these larger microplastics that are floating, the pump filter system can accomplish some different sampling goals. So not only can we sample a larger volume of water, we can also sample down to really small size ranges, so down to 10 microns in size, which is not super well defined yet in the research field, so we're really excited about this. We also can vary the depth that we're sampling because the pump filter system uses an intake hose. So we can vary what part of the water column we're sampling from. And hopefully, as you can see here in our density graphic or table again, begin to capture some of those higher density plastics that are sinking lower into the water column. So this image here of the pump filter system shows you a little bit of how it works. We have a gas-powered pump on another side, not pictured here, that's pulling water through the system from an intake hose. Water flows across 3 cartridge filters of different sizes. We have a 280 micron, 100 micron, and 10 micron. So really getting a robust kind of stratified sampling of these different size ranges. We also employ very strong contamination control measures to our pump filter sampling. You can see we also wear our orange, we put out our sign. We backwash the system beforehand using filtered seawater to try to blast everything that might possibly be in there out. And we take a control sample before we take our environmental sample. And we process all of those control samples in the same way as we would the environmental sample to get an idea of any background of contamination that might be present. So how are these processed? The pump filter system sample processing is a little more extensive than Mantitrol because we're looking at a much smaller size range and because we have a higher concentration of all kinds of debris that we've pulled in from our water body. So we start by sonicating the stainless steel cartridge filters that are in those kind of cups that were in the pump filter system. And that allows us to loosen anything that might be stuck in the pores of the stainless steel mesh. We sieve to reduce the water volume, and then they go into a digestion. So we use an alkaline digestion for 24 hours, and that's really to help reduce the bulk of the biological material and organic material that's present in the sample. This is really crucial because we collect a lot of biological material. And this is also one of the reasons why we can't just Mantitrol with a smaller net pore size because it would just get clogged so quickly. After digesting, the samples are density separated. This time we are using potassium formate for our density separation, which is a much higher specific gravity. And as you can see here in our setup, we have a funnel with a flexible silicone neck clamped closed to the bottom. And our samples sit in the solution for a minimum of 5 hours to allow plastics to float to the top. And then the dense stuff like our sediment and other things that we don't want to keep, we're able to drain from the bottom and reclose that clip without losing our sample. Final step is filtering everything onto a polycarbonate slide. And then we use a couple of different stains to aid in our identification of plastics. We use Nile Red, which is a really commonly used stain in microplastics research, and it creates fluorescence in a lot of plastic materials. And we also use a combination of Cockflower White and Evans Blue stain to help us stain any remaining biological material, which I'll show you in a photo in a moment, which is super helpful because the digestion technique gets rid of a lot, but it doesn't get rid of everything. So we can rule out any remaining biological stuff that we might have caught. So now our sample is on our slide. We can scan it into our Olympus microscope. And as you can see, we use bright field, so normal visible light scan, as well as a few different fluorescence channels. So this is under fluorescence. We can see a lot of blue, which is the biological material that's still there, and lots of other things. So now what do? How do we know possibly what is plastic on here? How are we going to identify it? From this kind of step forward, we can't use that melt test that we use with Mantitrol because our target particles are so small. There's just no way to have a hot needle that fine that's going to be able to melt these particles without destroying them. So we subsample our filter. We've decided to use a cake slice subsample. This is really due to the behavior of all of the particles on the vacuum. Things tend to accumulate towards the edge as we're washing things through the vacuum. So we want to capture those edge pieces as much as possible. So we use a cake slice and we can feel that that's representative of our filter as a whole. So within our cake slice, we identify potential targets, and all of these need to then go to the Raman. So all of our targets will get Ramaned, and then we can extrapolate from that subsample how many plastic particles are likely in our total volume of water sampled. So these are a couple images of some of the fluorescence that we utilize in the lab. So this is under normal visible light conditions. You can see there's some very obvious pink beads in here. These are spiked polystyrene beads. Not only are they pink in normal light, but they're going to be super pink under fluorescence channels. However, there's nothing else on here that would jump out to me under visible light as potentially being plastic, maybe a couple of the darker particles. But when we add our fluorescence channels, we can see that we've now really uncovered this hidden characteristics of these things that would otherwise be invisible. So our blue stuff here is biological, so we can rule that out. And then we can see this really brightly fluorescent green kind of fiber that I would consider definitely a potential target that we would then take to the Raman and see if it's fluorescing, or see if it's a plastic, excuse me. Contamination control in the lab. Again, we wear our orange. We do not have jumpsuits to the lab. We do have orange lab coats. And all of our sample processing, when humanly possible, is done under a laminar flow hood. This allows us to have samples uncovered as we're processing them without the fear of things from the air just falling inside, or anything from our clothing falling over our sample as we're working on them. The laminar flow hood brings the air down and out towards us. So this is a really helpful way to increase our confidence that what we're identifying as plastic truly came from our environmental sample, especially down to our really small size range. We use procedural controls, as I mentioned previously, whenever possible. We filter our reagents down to 0.2 microns. We don't want to introduce anything to our sample from any of the reagents we're using. And we also use ultrafiltered pure water whenever possible, whenever we're preparing or washing anything in the lab. Our last and probably most important of our procedural controls that we use are extraction efficiency testing measures. So we use known amounts and types of spikes. So known amounts of plastics, whether those might be, as you can see in this photo, beads or fibers or fragments, and run those through a blank sample, essentially from start to finish. So we can see how effective our methods are at extracting every single particle from our sample. So we'll use pure water and a mix of these different types of plastic spikes and run through the pipeline and see what we get. So in our lab, we aim for over 90% recovery, and we have been able to achieve that in all of our processes that I've mentioned so far. And this allows us to really make sure that any of the approximations of the amounts of plastics that we're saying are in the bay are really technically sound, which is super important because we don't want to make any false claims about things that maybe came from our lab, or if we have lower, say we have 40% extraction efficiency, we're losing a lot of stuff from start to finish that might be potentially very important for better understanding how our ecosystem is functioning with this pollutant. So I think it's time for a quiz. So we're going to do a little challenge here to maybe break some of the monotony of just me talking. Since we've talked about the methods, how we select plastics in the lab, I think that you all should have a shot if you would like. So I'm going to show you first a couple of images of some of those Petri dishes of our mantitol samples under a microscope, a dissecting scope. And you're going to see if you can spot any potential plastics that might be floating on top. So I'll give you a hint for the first round. They're probably going to be more identifiable from their color. That might be different from the other things around it. So are you ready? Okay. So we have two different slides here with two different particles that are present. So give everyone a moment. You can see we have a lot of stuff floating around in here. We have seaweed. We have some organisms as well. We have probably just like leaves and sediment. All right. Give everyone a couple more seconds before I reveal them. So in our first slide over here, we have a blue kind of teal microbead that's floating pretty much right in the center. And on our right-hand slide, we have a pink fragment. So these were both actual particles pulled from our samples that I snapped a photo of because I think it is really interesting to try to find them. don't worry, myself and our undergrads go through a lot of practice to find them. So if you didn't find them, don't feel bad, but we're going to amp up the difficulty here. So this is round 2, and these are really not going to be visible from their color as much. So they might be a little bit more cryptic. All right. Over here on the left, we have a film over here that's floating. And same on the right, we have a film that's caught in some of our debris that's towards the side of the Petri dish. So again, these are really cryptic. And you can imagine if I were an organism that's feeding on stuff in the bay, I'm never going to be able to visually distinguish between these pieces and the things floating around it. So if you got them all right, please let us know. We'll put you on the payroll in the lab and you can help us pick particles. So another quiz we're going to really quickly do is just to see if we can guess that polymer. So I've talked a lot about Raman spectroscopy and our ability to identify polymer types from environmental samples. And so this time you can have your hand at guessing what you think some of these might be. So here's our first one. This is also recovered from our Mantitrol surveys. If you want to put your guess in the chat really quickly, or if you want to shout it out, you're welcome to. So we're looking for the type of polymer that this might be. Give everyone a few seconds. All right. Let's see, we have some chat ideas. We have polyethylene, polystyrene. This one is polypropylene. So polypropylene, I often associate with being very colorful. Polypropylene is usually used in a lot of commercial plastics for lots of different types of things, but often has a dye associated with it to change its color. Round 2 is a little bit different, more of a fragment that's pretty flat. This one is probably a little bit more flexible as well. It's not going to be as hard as that polypropylene. This one is polyethylene. So this likely came from something maybe like a plastic bag. And last but not least, this one to me is more visually easy to identify. I think because it has a really a familiar structure, something maybe from like a takeout container of days past. It is polystyrene. So again, these are all fractions of millimeters in size, but when we take these really high resolution images of them, we can see all the facets of these particles that have been weathered and changed and biofouled over, we don't know how many years in the bay, which I think is really interesting. So thank you for participating in my quiz, and I'll get back to some results, which are very interesting. So this figure here is our Mantitrol outcomes. This is all preliminary because we are still sampling. We have a few more sampling events to complete before we can finalize our data. So this is showing on a north. So over here is our north end of the bay to south gradient, and I'll pull that map back up for you in a moment. What the seasonal variation of the amounts of particles recovered looks like. So we can see that we have pretty high variability between seasons, between space and time, and within sites, and that generally we recover more particles closer to Providence, so in the north end of the bay. We see some seasonal patterns emerging, but we want to definitely make sure that we complete our sampling before we make any kind of outcomes from this. So this is our map again, just for reference. So we have CPO up here, we have HOG, west of Hog Island over here, and these are our 2 highest count sites. Andy also shared a map of the bay that had some locations of some areas of interest, including some wastewater sites as well, which could potentially be contributing to that higher concentration of particles in that area. So if we compare this outcome to some other areas in the United States, so Narragansett Bay up here, our average concentration across all seasons and all sites is about 0.3 particles per meter cubed. The Chesapeake Bay, on the other hand, is a little bit lower than that. And then on the high end over here, Monterey Bay is at 1.32, which is pretty high compared to our average and maybe matches some of our highest concentration samples recovered. So we can pretty confidently say so far that Narragansett Bay is probably in the middle of the amounts of particles per meter cubed. But it'll be interesting as more studies come out using Mantitrol methodology to continue to compare and really see where we sit within that strata. So some additional data that is just interesting on the breakdown of the types of particles that we're covering. We've recovered well over 2,000 particles. So again, would be impossible to Raman all of those, which is why we subsample. Again, more particles in the north, and we definitely want to continue that seasonal sampling. So I'm going to break down what some of these look like. So you may have recalled that Colleen described that a lot of the research points to fibers as being the most populous pollutant type of microplastics found in marine debris. That is not the characteristics of our sampling, but likely because we are sampling surface water. So a lot of those fibers that are made of denser materials are going to be sinking out of the top of the water column. So we see a small percentage of beads, mostly fragments, and some fibers. The fibers that we do recover are mostly bundled polypropylene fibers. These are some examples of the images that we've captured. Colors are interesting as well. We see mostly clear and white, but our next most populous color is blue, which I think is interesting, as well as black, which could be coming from car tires or similar substances, green, pink and red, yellow, and tan or brown. Polymer types, we see predominantly polyethylene and polypropylene. We see a little bit of polystyrene and a healthy amount of other types of particles as well. Some of these are other, we know it's plastic, but we're unable to get a clear signal on what type, or just other types of polymers that we can identify, but just weren't broken out for the purposes of this graph, just to show the overwhelming amount of polyethylene and polypropylene that we're finding. And these are some examples of the Raman spectrographs for each of these types of polymers. So we're really interested in making some further investigations onto the influence of tides in the amounts and types of particles recovered in Narragansett Bay, since it is a tidal system. So we actually did some kind of short-term sampling to do some investigating about how this might be changing what we find. So we implemented 2 of what we call high-frequency sampling events in the east and west kind of southern parts of the bay, where we sampled, instead of just doing one 10-minute trawl and moving on to the next site, we trawled 10 times over the tidal cycle. So starting at high tide through low tide and back to the next high tide. And what we found was pretty interesting. In our first sampling event, we see a pretty clear connection between the tidal height, which is this line here, and the amount of particles recovered. This doesn't hold as true in our second sampling event, so it would be interesting to continue investigating this tidal connection, because we know obviously these different sites in the bay might be influenced by different oceanographic variables, so they're not mirror images of each other. However, this is important when we're planning for additional sampling, because it means we can't just drop in at any point in the tidal cycle and do sampling and compare it to another site, to another day, potentially. We need to make sure that we are considering the influence of the tides on the amount of particles that are being recovered. Last but not least, our very last upcoming project that I'll really quickly describe is our trophic transfer experimentation, which are going to be in URI's mesocosm facilities, which are very neat facilities that have 10 tanks that are adjacent to Narragansett Bay, but allow us to manipulate some variables that we would not be able to otherwise manipulate in the bay itself, such as adding plastic, and check out some different outcomes. So Colleen described an experiment looking at Eastern oysters and their resiliency to polyester fibers. And this study is going to take that one trophic level further. So we're going to have Eastern oysters and we're going to have blue crabs, and basically assess not only the ability of or the behavior of plastics as they're potentially moving up in this food web, but also the impacts on the organisms themselves. And we're really excited to kind of continue building this out and launch this later this summer. So that is all I'm going to talk about our projects for now. I know my last kind of core piece, as we're running out of time, I'm sure, is how can you get involved? So I wanted to call back to a slide that Colleen shared earlier in the presentation with some really wonderful resources from lots of different organizations that work with local education and outreach, awareness, cleanups, and I think who are experts in communicating to individuals actions that they might consider taking to make a positive impact in the plastics crisis, particularly when it comes to marine debris. I think that individual and collective actions are highly dependent on a lot of different variables, and they need to be equitable and accessible. And I think that these organizations, especially ones that are local, have a really good idea of how to communicate those in a socially relevant way, so that lots of people can partake in the movement to be more environmentally conscious. So if you would like links to any of these, please let us know. We're happy to provide them and also point you in the direction of some additional ones. From a research standpoint, we are really interested in getting more people involved in marine debris research. For example, things like community science. So we're really interested in continuing to explore that as an avenue. I developed a program called Mapping Microplastics, which uses a pretty pared down version of our methodology to engage community scientists of all ages and backgrounds in testing their local waterways for microplastics. And pretty simple community science methods. We collect, we filter, we stain, and then we look for fluorescence. So tapping into those fluorescence techniques. So these are the kinds of projects that we're really interested in pursuing, and hopefully adding to the collab repertoire, or just sharing the techniques with lots of different kinds of people, teachers, organizations who want to get more involved in some hands-on science research. So I will yield my time back to Andy, who will close this out.
Thank you very much, Sarah. So yes, we are sometime. I thank the 65 of you who managed to make it to the end there. And I think we're ready to take any questions. So thank you very much, everyone, for listening.
Great. Thank you, everyone. I just wanted to, if you want to raise your hand or if you want to ask your question in the chat, please feel free to do so. And actually, we have a few questions in the chat, but since I'm the moderator, I kind of want to take advantage and ask the first question, because one of the things that always struck me about the work that you guys do with plastics, especially microplastics, is measuring sizes, right? So there's this difficulty, it seems like you can only measure to a certain, I guess, particle size. And Sarah, you had mentioned kind of, I think it was the pump filter system that has allowed you to sample smaller sizes than have typically been recorded. So one of the questions I had was, Colleen, when you had, brought up one of the figures about 8 million tons of plastic in the ocean, is that do you think that's an underestimate, considering we haven't really been able to measure and quantify these smaller particles?
Yeah, this is a great question. Thank you, Meredith. I'm going to give a complicated answer because I'm a researcher, but it's a bit of both. For the larger materials that we have a better handle on being able to physically see easily and collect easily, it could be like an underestimation, because also we're losing materials that are sinking through the water column. And at the same time, it could be an overestimation depending on like where you're collecting. Like Sarah showed that nice schematic about how you sample and where you sample and when you sample has importance as well. So these are some of the questions that we're still trying to better understand so that we can improve the global community's like methodological approaches towards quantifying this. With respect to microplastics, there has been some studies suggesting that the microplastics estimation in early studies was overestimated because people were not doing a lot of control for contamination practices, which meant there was a lot of contamination coming into their samples, which they hadn't necessarily focused on or tried to avoid, and therefore contributing to their sample. But researchers globally are doing much, much better on that now that we're getting the global community to learn more about how we can do better on those approaches. One of the challenges, like Sarah said, is that there is no global standard method which everyone is using. It depends on what facilities you have, what infrastructure you have. It's not a cheap area of science to study in, and we've Both Andy and I have kind of pulled our startups together to be able to come up with some of the facilities to be able to do this work. But people are learning more about what the best practices are and adopting that, which is what we're doing through conversations through some of the audience like CAHO and EPA. I know they've been doing as well. So as long as people are kind of are using the kind of latest understanding about what the best practices are, then I think that's a movement in the right direction to getting at more accurate numbers.
Great, thank you. And CAHO had actually asked earlier what percentage of particles are confirmed with the hot needle method that are also confirmed as plastic using the Raman method. And actually, when you were saying about the hot needle method, I was like, that must be so tedious. Do you have to go through every single particle? Because yeah, that sounds very tedious.
We do hot needle every particle from our Mantitrol work. It's tedious, but we've worked it into the workflow, I think, in a way that works. When we interview potential undergrads, we make sure they understand that some of the lab work might be repetitive, and they're very eager to help. So fortunately, we have a very wonderful team helping us get through that tedious stuff. And to answer the question of how much or what percentage of what we hot needle test that melts is Raman and confirmed to be plastic, I don't have that exact number, but it's very high. I would say the only thing that I've ever seen that we have melt tested that has melted and was not plastic was wax beads. And that's very infrequent. So I would say that number is pretty close, in the high 90s, if not very close to 100.
Great, thank you. And there was another question about kind of concentrations found globally. Would heavily industrialized coastal regions, would you think would have a higher concentration? And I guess because you've compared bays, estuaries that are somewhat protected from the ocean, because if there's no global standards, I guess I'm also curious too how the transfer with circulations either between states along the US coast or even countries and how that might work. But I think to this question, it was kind of like, how do you see the variances and concentrations?
I think I can take that one. I mean, the biggest challenge for us in comparing to different locations is everyone uses very different methodologies, very different processing pipelines, very different conversions. Some people don't just report the total number of particles with no assessment of how much water they've actually sampled. So cross comparison can be almost impossible. I don't know if Sarah, if you have any other Mantitrol surface water numbers from industrialized areas?
Not exact numbers off the top of my head. I could certainly pull them if people are interested, and we can send a follow-up. But it definitely is higher in more industrialized areas. I think there's been a lot of Mantitrol dirt work done in China, and they're pulling some pretty high numbers from there as well. There's not as many studies done in the US, which is why we saw there's pretty high variation in those three. But again, echoing Andy, we're not always using the same techniques in the lab. This is a field that's still trying to standardize the processing protocol. So I think there certainly are trends, and it would follow that more industrialized, urbanized area are going to have more plastic, but it's still a question mark, I think, kind of as a whole right now.
There's a question about the likelihood of seagulls dying from plastic indigestion, which I guess you could they're asking specifically about seagulls, considering they've seen a lot of dead birds around their workplace that is near the water, but thinking about that slide with the sperm whale, and we've also seen other marine animals, kind of their stomach contents. So I don't know how you would answer that question, but I guess I'm curious, do you have an idea of the likelihood, or is that still like another, like more ongoing research for that?
Well, Sarah has posted a very, very helpful link into the chat, which gives some fairly good meta-analysis numbers of that. I mean, ingestion is very common. Organisms are not used to non-breakdownable material in their gut. So, I mean, Sarah, do you want to add to that? But that article that Sarah said gives the answers to Jen's question.
I actually feel that this might, Colleen, you probably have about individual organisms or different types of organisms and kind of what the outcomes might be.
I was searching for something in the meantime. Can you just rephrase the question? Sorry.
So what is the likelihood of plastic ingestion harming or adding to mortality of seagulls? And actually, I'll add to that because it seems like there's two potential impacts. One that's physical blockage, and the other one, what you described as toxicity. And I'm kind of curious between the two, but the primary question is, what is the likelihood of seagulls dying from ingestion?
I mean, our focus has been mostly on like marine invertebrates, so we couldn't say for sure, but there has definitely been entanglement. I mean, in the media we've recently seen masks being like tangled up in like seabirds' feet, and we know that there are some seabirds out there which are actually being used as an internationally recognized bioindicator for plastic pollution. So I think it's the northern farmer. Someone might correct me if I'm wrong on the name of that. But there is a seabird in the North Sea, which most of its life, it lives out in the open ocean. And these have been used to, when they find dead birds, whether they've died from plastic ingestion or not, I can't say right now, but they have plastic presence in their stomach in most cases. And these are from, like, because their lives are mostly on the ocean, they can use this as an indicator for like ocean plastic ingestion from animals. So there is a lot to learn still. I know some researchers in GSO, the Graduate School of Oceanography, have been doing some work on like plastic presence in seabirds, but there is definitely evidence to show that they are ingesting them, and it probably is going to be causing digestive, some physical damage to those animals for sure, at least.
Okay, great. Lou Rothstein has a question. So Lou, I'll let you go ahead.
Okay, thank you, Meredith. Great job, Andy, Colleen, and Sarah. Thanks for this wonderful group of talks. Colleen, you mentioned that most plastics do end up in the sediment. Do you guys have any plans on using the mesocosm to look at some of the potential biogeochemical and ecological impacts of plastics in that environment, in a controlled environment?
In the sediment specifically, no, but I know that there are people in the audience today who might be able to comment on that. I know Kay Ho, for example, and Tom Boving have been measuring sediments plastic presence and found some phenomenally high numbers in sediments in Narragansett Bay. But yeah, it's a good question. For us, it's more about looking at asking some of the basic questions first before we can make that leap into understanding what that looks like. But it's definitely, you know, things we do need to look at.
And a follow-up to that, when you're looking at concentrations of these plastic in the sediments, you're looking for convergence zones in the physics, where the physics might converge these materials and then sink. And there are known locations in Narragansett Bay that are kind of like little gyres that do converge those particles. So it may be useful to have a look at some of the work that Chris Kincaid has done in that, using the circulation models that he's developed over time. And he does identify some convergence zones that may point you in the direction of sampling sediments in those locations.
Yeah, I'll let, I think Andy will have a response to that.
I do, and thank you for that, Lou. Yes, we are working with Chris Kincaid and the ROMs and OSTOM models to try and predict where plastics are going from source points. We've also tried to do some validation of that with drifter data sets from Scott Rutherford and Roger Williams. So we're trying to bring that hugely complex, hugely noisy data set together with our plastic sampling data set. But you can see from what we saw with our tidal sampling that it's just orders of magnitude more particles coming at different parts of the tide. There's so much going on with dynamics that's going to take, I think, a few years and some real fine experiments to try and figure out really what's going on. But yeah, we are thinking about that.
It's a challenge. Good. Thanks, Andy.
So I'd like to add another question about kind of why the choice of oysters. And I guess since they're filter feeders in your studies so far, are they able to kind of process out the particles maybe they've ingested or anything like that?
Yeah, this is a good question, Meredith. Thank you. Yeah, so there's been work done from researchers at the University of Connecticut, which have showed extremely great selection capabilities of filter feeders to basically passively sort through particles as they kind of whiz past their tissues, so they can choose whether they ingest them or whether they reject them as particles. So there is already some like good selection going on by those. But again, what we're really just trying to elucidate really is trying to address some of those basic questions. What if under these kind of particular environmental situations will happen, if anything, to these particular animals? And the reason we focused on this particular species is because previous work has shown, has focused on other types of oysters and not necessarily this species, the eastern oyster. And some of the work that's come out from our group has shown that responses to plastics is species specific. So it's definitely worthwhile trying to examine more closely how all these different species are responding so that we can get a better idea about what's going on.
Great, thank you. Do you guys have any future plans maybe to kind of compare oysters with clams or quahogs or mussels? Just curious as kind of like from an aquaculture standpoint, the various species that people might be curious about.
We think it'd be really interesting, and we would love Sea Grant to fund work like that. Yeah. So yeah, no, I think, this is a great question, and there's a lot we still don't know, and there's a lot we still need to do.
All right, I'll ask one more question, kind of back to the technology and resources at hand. What, I guess, would you like to see? Do we have technology at hand that can measure kind of particle sizes that are smaller, or what do you think the next step for the scientific community is to kind of better assess the array of particles that are entering the ocean?
I can take a first stab at this one. I mean, one of the biggest challenges we've got is the concentration of microplastics. Is how do you go from 1 cubic meter to 1 particle? And how do you handle that volume of water is where the challenge really is. There needs to be some approach to concentration of potential targets prior to some analytical approach. So in situ monitoring is a challenge, particularly in water samples. There are some approaches that might be emerging that could be used for stratified size range centrifuge or density separation centrifuges and things. I think there needs to be, that is an area of intense interest to try and maximize what we can do and minimize the false targets, minimize the contamination, and improve the amount of sample that we actually have access to and can image, can quantify, and so on. So in situ sampling, I think we are some distance away from yet, unless you are in an extremely polluted area. Sarah can speak to the logistical challenges of working with it. And I want to stress, I hope it was clear, microplastic research is not for the faint of heart. It is very technically challenging, very logistically challenging, and very drawn out process. But Sarah, do you want to add anything to that?
No, I think you described really well kind of what the goals might be of what that technology could look like. Devices that could automatically count the number of particles that are flowing past some kind of sensor that could be developed. Like Andy said, it's probably not something that's going to be developed in the next couple of years, probably maybe 5-10 years out from that, I would guess. And again, as Andy mentioned, right now, we're really at the point where without isolating the particles, we really can't confirm that they're plastic. That's kind of where the state of the research is. And with every publication, we're improving this methodology, which is really cool. It's been awesome to see how these methods have kind of exploded in the past, 10, really 5 years, as more and more people have developed different techniques. But it will be really cool to see kind of what that next step is in being able to quantify those smaller particles. But it just kind of brings to mind the magnitude of how simple it is, what a simple act it might be to litter a macroplastic, but how difficult it is later to identify the impact that has had. And that's kind of what I always return to as we're trying to sift through these particles in the lab.
Great, thank you so much. I don't see any other questions in the chat. And so I want to ask Andy, Sarah, and Colleen, if you guys have any other final remarks you would like to make.
Well, I think we've been talking for quite some time, so I think people might want a break, but I just hope if you were interested in the research we're doing and you want to learn more, check out the AutoMap website or reach out to Colleen, Sarah, or myself and ask any questions you may have. We're really interested in looking for engagement with stakeholders, and if people are interested in working with us, just drop us a line. We really appreciate your time, and thank you very much, everyone. I don't know if Colleen, Sarah, do you want to add anything?
Just want to say thank you very much. It's been great to speak with you and have this opportunity to share what we've been doing, and we're really excited to learn more about what others are doing across the state, across time. So thanks very much.
Great. Well, thank you to our speakers, and thank you again to our co-sponsors for today. And we want to thank everyone who joined us this afternoon. So enjoy the rest of your week and take care, everyone.